Welcome to Speaking of Oats. I am Jim Bradeen. I'm a professor of plant pathology at the University of Minnesota, and I'm the administrative director of Oat Global. I'm joined today on this webinar by Charlene White, who will field questions for us, and by Grant Zadzik, who is keeping everything moving smoothly. As you might know, Speaking of Oats is a community forum for anyone and everyone who is engaged with the oat industry, oat research, and oat production. And this series is a partnership between Oat Global and the Oat Newsletter. And we're very happy that you are here with us today. Now, before we get started, there are a few housekeeping items I want to highlight. At the bottom of your screen, you will find the chat function and the Q&A box. If you have questions for today's speakers, please use the Q&A box. For all other items, please use the chat function. And we'll have time today at the end of today's webinar to address some of the audience questions. All right, so today we have something really very special for you. We will hear about the USDA-ARS Oat Grand Challenge, and we have several panelists. And I'm going to turn it over to José Costa, who is the National Program Leader for grain crops with the USDA-ARS. Jose, thank you and your colleagues for being here today.
Thank you, Jim. This is Jose Costa, and I wanted to introduce the whole team. I'm a national program leader for grain crops at USDA-ARS, and also today we'll have a stellar group of presenters that will include Kathy Esvelt-Klos from Aberdeen, Idaho, Jason Fiedler from the Genotyping Lab in Fargo, Shahryar Kianian from the Cereal Disease Lab, Jay Ohm from the Phenotyping Quality Lab in Fargo, North Dakota, Dave Garvin from St. Paul, Minnesota, Sharon Schneider, some of you may know her as Sharon Papernick, her previous last name, and from Brookings, South Dakota, Jean-Luc Jannink from Ithaca, New York, Tanner Shen from Albany, California, and Jim Harnly from Beltsville, Maryland. Okay, so next slide, please. Okay. So today I will present our research efforts on what we call the Oat Grand Challenge in USDA-ARS. And this is a special project created within ARS to increase collaboration and synergies for research. And in this case, we are focusing on oats. Next slide. So can you go, let's see, I think you're one ahead. There, so oat is a valuable crop, a very valuable agronomic crop, and it has several benefits. It lowers LDL cholesterol levels, reduces cardiovascular disease risks, and provides glycemic index stability. But the US is a net importer of oat for human consumption. Therefore, we need to increase the production of high quality oat for human consumption in the US. So that's our overall goal for this project. Next slide. So the question for us is, how can USDA-ARS help expand US production of oat for human consumption? And we're focusing on several aspects of oats. Mainly, it has to have higher yield, increased yields, also a better disease resistance, enhanced disease resistance, high quality. We also need to focus on production practices as well as nutritional quality. So all of these aspects we're taking into account to develop this Oat Grand Challenge. Next slide. So we have developed these 8 objectives within the Oat Grand Challenge in USDA-ARS that include developing improved oat germplasm or pre-breeding, especially at Aberdeen, Idaho, and the rest of the objectives are listed here. And also I want to point out that we will expand this pre-breeding or even further with the hiring of a new breeder at Fargo, North Dakota. So now I will introduce Kathy Esvelt-Klos, who will take over from here for the next slide. Thank you.
Okay, thanks, Jose. I'm part of the small grains and potato germplasm research unit at Aberdeen, Idaho. That's the red dot on the map that I show there. We house the National Small Grains Collection, which is led by Harold Bockelman, and right now has available over 10,000 Avena accessions. And we also have the Barley and Oat Improvement Project, which has 3 full-time scientists, myself, Gangxi Hu, who's a breeder, and Dongying Go, our new functional geneticist. Next slide. I'm the pre-breeder, so I'm mostly going to be talking about pre-breeding. In my program, we investigate the genetic architecture of oat quality traits and also oat agronomic performance, but we spend quite a bit of time pre-breeding for disease resistance, and most of that time is spent on crown rust and stem rust resistance for the reasons that are outlined in this slide, but Shahryar is going to talk a little bit more about that later, so I'm just going to move on to the next slide. Like many pre-breeding programs, we have a 4-stage approach. The first is to identify accessions that carry traits that we're interested in, but which are not adapted for modern production. The next is to characterize those both phenotypically and genetically, then to map the QTL that underlie the phenotypes that we're interested in, and finally to deploy those QTL into adapted germplasm that we can move along to breeding programs. So for example, crown rust resistance, we started by surveying the National Small Grains Collection, and we identified 2,048 Avena sativa accessions that had not yet been characterized for their response to crown rust in the field. And so we sent those down to Steve Harrison's program at Louisiana State University for screening. The ones that looked promising there, we rescreened in the Matt Moore Buckthorn Nursery in St. Paul, Minnesota, and then we repeated that a couple of times. And the ones that we were still interested in after that process, we took into the growth chamber to characterize for whether or not those accessions carried a seedling form of resistance or an adult plant form of resistance or some combination. So now, after screening over 2,400 Avena accessions for both crown rust and stem rust, we've identified a number that we're interested in moving forward into the mapping phase, and we're currently developing populations for that. But while we're waiting for those to be ready, we're collaborating with a number of other institutions around the world, really, on mapping the locations of previously deployed resistance genes. So for example, we were part of Ida Kabiti's paper where she published the location of the stem rust PG13 gene. Next slide. This slide outlines our efforts in population development towards mapping quantitative adult plant crown rust resistance in the accessions that we've identified out of the National Small Grains Collection. So the list of accessions on the left are those that we're interested in with the notation on the generation that our population is in currently. They've all been crossed to the same elite breeding line out of Gangxi's program. And what we expect to find is that for each of these accessions, adult plant resistance will be conferred by numerous QTL, each with small to moderate effects on phenotype. But that if they go according to what we've seen in the past, many of these QTL will be additive in their effects, such that we can combine the most promising QTL from numerous sources of resistance into a single germplasm line. In order to do that, or to guide that process, we need to know which of these QTL are unique from one another, but also unique from the QTL that are already available in elite oat germplasm. And so to answer that question, we've also been working on the populations that are kind of outlined to the right of this slide. And these are crosses with existing lines that we know from their pedigree derived from one or more of these 4 traditional sources of adult plant resistance to crown rust. Next slide. And this final slide is just a word on the breeding program here at Aberdeen. Gangxi, whose program focuses mostly on agronomic and quality characteristics because disease resistance is not a big problem in our particular production area. And as you can see, he has numerous advanced breeding lines that consistently outperform his checks in our production conditions. And so with that, next slide, I will turn this over to Jason Fiedler, who will be talking about establishing high throughput genotyping platforms.
Thank you very much, Kathy. Thank you, Charlene. Good morning. My name is Jason Fiedler. I'm a research plant geneticist with the USDA-ARS in Fargo. I run the small grains genotyping lab, and we operate as a genotyping service provider for the small grains community. My role in the Grand Challenge is to come up with high quality, high throughput genotyping platforms to support the various association studies that folks are working on for marker discovery and for marker deployment to quickly use all this information for germplasm selection and for breeding. Next slide, please. So this is a very nice image of the oat genome from Rick Jellin at BYU. For the non-genomics folks in the audience, oat is a hexaploid, meaning that its genome consists of 3 similar subgenomes, each with 7 groups. Now at large scales, oat acts like a diploid with 21 chromosomes. However, at smaller scales, there's a lot of repetition throughout this genome, and any given location on one subgenome looks very similar to another, which makes genotyping quite difficult at times. Next slide, please. So the largest challenge for my part of this project is developing nice, clear, co-dominant markers. These images here represent the popular genotyping platform, the Illumina 6K. I run this in the lab for oat a lot. But really these images can be extended to any of the assays that we end up running. So what we're looking for any design is the far left plot, where we get really nice, well-resolved clusters that represent 2 homozygous alleles. Now, unfortunately, what we normally see with many of these assays is the middle graphic, where you have at least one artifactual heterozygote cluster caused by the overlap of 2 different sites, even in things that are well inbred. And what we really don't want to see is the far right graphic, where you have lots of different clusters kind of all over the place. These markers are quite difficult to call correctly, especially in high throughput, and are generally just kind of tossed out before anyone uses them for anything. We can't really go into details here about why having a good reference sequence helps, but it really goes a long way in helping to tailor either the probes that we end up using or the analysis pipelines to help develop more of the good markers on the left and less of these marginal ones in the middle and on the right. Next slide, please. So what are we actually running in the lab? In oat, there are 2 main platforms that we run. The Illumina 6K assay provides genome-wide markers with full data coverage, which is nice. However, many of these markers are not very useful due to the multiple clusters kind of all over the place. GBS is another nice technique to identify novel genome-wide markers, but the data coverage is generally quite sparse. The Rapture edition that AAFC has developed helps make these markers more useful, but the licensing issues of GBS limit my ability to really help provide this as a service. So moving forward, I'm currently working on 3 new platforms. One is a new and improved Illumina array, or potentially a set of arrays. The advantage here is that we have a lot more genomic information than we did before, and we can pick a lot better sites for probe design, ones that are going to give us those really nice, clear markers. Another platform is single primer enrichment technology. This is a commercial product from TCAN, and it's worked pretty well in other crops for lower density genotyping. Now, for an initial study that I just ordered, we're targeting 2,500 SNPs that are informative in the various oat breeding programs that we support. And we should have an idea of how well this works once the reagents come in and we get them tested. Finally, allele-specific PCR like CASP or PACE is not really a new platform at all. It's very commonly used in all the other small grains, but has not been used in my lab for oat previously. So we started doing this. We brought in a few dozen of these markers that are associated with various traits. A lot of them from Kathy's program for crown rust, some other ones for yield and quality from the breeding programs. And these just need to be validated, and they'll be available to the community very soon. Next slide. So finally, I just want to thank PepsiCo for releasing the first reference genome to me and making my job quite a bit easier. I also want to plug at this point the global Pan Genome Project, the Pan Oat Project, which is taking this effort a lot further with providing 30 genome sequences to the community. So thank you for your attention, and I'm going to turn it over now to J-Bomb Ohm for high quality phenotyping.
Hi, my name is Jay-Om, and I'm working at USDA-ARS Hard Spring and Durham Wheat Quality Laboratory at Fargo, North Dakota. We evaluate quality traits of the oat samples and provide the data to oat breeders. Plant breeding programs usually require efficient, rapid, and cost-effective evaluation of many samples in compressed time. However, quality evaluation is usually time-consuming and labor-intensive. Therefore, it is essential to develop high-throughput phenotyping platforms to facilitate screening of high-quality oat genotypes. Next slide, please. The key factors to claim the health benefits for the oat are beta-glucan and total dietary fiber contents. The FDA listed whole oat growth flour as an eligible source of soluble fiber. Whereas it is very important to screen high fiber oat genotypes. However, the analytical procedures of beta-glucan and total dietary fiber are very time-consuming and labor-intensive, and this caused delay in evaluation of the beta-glucan and fiber content for oat samples. Next slide, please. One research in my lab aims to develop enhanced prediction models of beta-glucan and total dietary fiber contents using FT-NIR and FT-MIR spectroscopy. Recently, there are developments in AI algorithms. I plan to apply machine and deep learning algorithms to calibration of the prediction models of beta-glucan and total dietary fiber content. The improved prediction models will help evaluate accurate and rapidly the quality traits in oat. Next slide, please. Oat globulin fraction, which is also called abenalin, is the major storage protein fraction. This protein fraction composes about 70 to 80% of total proteins in oat. The higher content of the globulin in oat protein is also associated with the higher nutritional value of oat protein, as this protein fraction contains higher levels of the essential amino acids, such as lysine and threonine. However, oat globulin has poor solubility in water, and this limits the food use of the oat proteins. Despite the importance of the oat globulins, no report has been reported on variation of globulin protein profiles for US oat genotypes. Next slide. Therefore, my next research plan is to develop an improved liquid chromatography procedure to characterize proteins in oat. Specifically, this research object to enhance the size exclusion HPLC procedure for analysis of oat globulins and provide information on their environmental and genotypic variations. In this research, we plan to apply UV-RI and mole detectors to characterize the protein fractions. This research is expected to improve the speed and resolution of oat globulin analysis and contribute to screening of oat lines based on protein composition. Next slide, please. We also plan to evaluate other oat quality traits. The traits will include whole oat and growth characteristic measurement using an image analysis system. And we also plan to evaluate traits such as growth hardness, oat twirling characteristics, antioxidant flaking, and beverage making characteristics. Next slide, please. This laboratory also actively collaborate with other research groups on oat quality. And as a result of the collaboration, we published a paper. Yeah. Next slide. Yeah. Our next presenter is a renowned plant geneticist, Dr. Shahryar Kianian, and the title is Develop Genetic Resistance to Emerging Biotic Challenges.
Good morning, everyone. My name is Shahryar Kianian. I'm from the Cereal Disease Lab in St. Paul, Minnesota. I'll be talking to you today about the oat crown rust work, but not to exclude Dr. Eugene at Cereal Disease Lab, who works on oat stem rust. As almost all of you that work on oat know, oat crown rust is the most widespread and damaging disease of oat worldwide. Moderate epidemics to severe epidemics happen almost annually, and it can lead to yield losses of 10 to 40%. For example, in 2014, Minnesota reported 50% yield losses, and South Dakota reported 35% yield losses due to these problems. Next slide, please. What we know from the oat surveys that we conduct at Cereal Disease Lab on an annual basis, that oat crown rust virulence has been increasing. And as you can see in this graph from 93 to 2017, the virulence has been increasing, and it seems like pathogen has no problem accumulating various virulences with low apparent cost to the fitness of the pathogen. No negative associations between it. And there is lots of opportunity for sex and recombination with oat crown rust, because buckthorn is an alternate host who is a VD species that spread almost throughout most of the northern part of the United States and slowly creeping down to the southern part. There are over 100 PC genes that have been identified in oat, but their effective lifespan can be 3 to 5 years in the United States, and in some places it can be even much less than that. Next slide. So we have been focusing on two different aspects of oat crown rust. Is one, looking at the genomics of the pathogen to find out what is happening in the pathogen, what's leading to this virulence, trying to identify candidate effector genes. And there's a number of publications that came out. We essentially were able to sequence a number of isolates, and we did a small population study, and now I have expanded to a larger population study to look at what is happening and in the pathogen population, and also to identify possible effector genes by looking at association in the pathogen of the SNPs that are identified with various virulence races. And we can see that we have identified a number of candidate effector genes. Next slide, please. At the same time, we are continuing the work that was started by Dr. Marty Carson and his technician, Jerry Oshevsky, before I joined the Cereal Disease Lab, which is to identify adult plant resistance in oat populations. They identified about 9 different accessions of oat, Avena sativa, that showed adult plant resistance through multiple screening in buckthorn in the greenhouse and making sure that they are not single gene seedling resistance. Marty and Jerry developed F9 to F11 recombinant inbred populations, and we have been essentially analyzing those populations. We, meaning my graduate student, Eric Nazareno, who's been analyzing these populations, looking at this variation in resistance and doing the mapping. Next slide, please. And Eric has been able to identify a number of QTLs that are associated with adult plant resistance. And the green ones are the ones that have been verified either through following population studies or through a different population studies. And what we have been doing is developing CASP markers, link CASP markers for all of these QTLs. So we can pass them on to the genotyping lab and Jason for using in selection. And next slide, please. And through a scheme, we have been pyramiding these resistance genes to essentially be able to release those to the breeding programs, all breeding programs, with only caveat of keeping the pyramids together because the associated markers are available. Next slide, please. to essentially use in their breeding for developing more durable, resistant oat crown rust lines for study. And this is a picture of a couple of those pyramided lines that show very good resistance in a buckthorn nursery. So it is my pleasure now to pass it on to Dr. Dave Garvin from the Plant Science Research Unit in St. Paul, Minnesota, who will talk about genome editing.
Thanks, Shahryar. Glad to be here. I'll be talking about work that we're doing relating to developing a gene editing platform for oat improvement. This work is being led by Dr. Sarah Blecka down on the bottom right there, hiding behind her mask as we all are doing. Can we go to the next slide, please? So oat genomics has kind of lagged behind that of wheat and barley, but we're definitely picking up pace here. And I think we can benefit by having an entire spectrum of genomics and biotechnology tools for oat improvement. And that's really what we're interested in doing, kind of bringing genome editing into the whole mix for oat improvement. There's a couple basic things I kind of think about that we need for gene editing. We need an efficient agrobacterium transformation system. Oats have been transformed quite a bit through biolistics, but that's not really acceptable. It's too complex, difficult to get rid of. We need to be able to recover transgenic plants from our transformation. And then lastly, having a genome sequence is very useful. For instance, for looking at sequences of genes that we might wish to target for editing. And so I want to go on to the next slide. One of the challenges with oats is it doesn't really seem to regenerate well. And we've played around with a couple of different genotypes. We've settled on our work with experimental line called GAF Park. And this was developed by our longtime colleague, Dr. Howard Ryan, some decades ago. And it regenerates extremely well. You can see that on the slide here, or in the image, the cali and the shoot regeneration from them. It's been used previously for bialistic transformation. Can we go to the next slide? We weren't aware of any work demonstrating that this could be transformed with agrobacterium, but we've now shown agrobacterium is highly efficient in this line. If you look at the upper image, that's a set of immature oat embryos stained for GUS activity after co-cultivation with agrobacterium. And you can basically see nearly every one of these has GUS stained sectors indicating transformation. And now we've been playing around for quite a while now to figure out what we need to use as a tissue for transformation, whether we use immature embryos or on the right side, do we directly do callus transformation where you can kind of see some GUS sectors, but we've settled on using stem segments from young seedlings, and we just isolate those from seedlings like are shown on that lower image. Can I go to the next slide, please? So I think that we're getting close to having the entire package available for our transformation work, right? We have a readily regenerable genotype. We can transform that. We just need to now be able to demonstrate that we can get transformed plants regenerated after transformation. And on the right, the image here just shows callus being formed off of a transformed oat stem segment on selection, which would indicate successful transformation. We have material like this going on to rooting shortly, but I do feel like in a very short period of time, we're going to actually have all of the pieces together that will allow us to actually then begin moving into actually testing gene editing in oats. And I think that this will be a valuable tool for the research community as a whole. So with that, I'd like to pass the presentation on to Sharon Schneider.
Thank you. So I am Sharon Schneider. I'm the research leader of the North Central Agricultural Research Lab that's in Brookings, South Dakota. And also on the call today is the main scientist on our part of the project, that's Patrick Ewing. He's a brand new research agronomist in our unit. So our component of the Grand Challenge is a critical agronomic piece, and it's to place oats within the context of cropping systems. And for those of you who don't know, Brookings is in eastern South Dakota. It's within the primary oat production region of the United States. Our lab's overall research program focuses on sustainable cropping systems, and our scientific expertise emphasizes soil biology and entomology. Next slide, please. So our contribution to the Oat Grand Challenge has three components, and one is to help in the oat breeding effort overall by evaluating the field performance of advanced oat cultivars under varying field conditions. And we're particularly interested, as you are, in their tolerance to drought, to heat stress, and to pests. And we'll partner with the other labs that are in the Grand Challenge on this objective, and we'll assess yield and other productivity measures, and then we'll depend on others to evaluate grain milling quality, human nutrition traits, and disease resistance. Next slide, please. The second component of our oat research program will be to understand how biotic and abiotic agronomic, soil, and climate factors influence oat development. And here we'll have an eye toward optimizing nutrient management and better understanding relationships with the rhizosphere biological community and how those translate into oat productivity overall. This will be primarily conducted on our 150-acre research farm in Brookings, which allows us to do research under carefully controlled conditions. Next slide, please. And our stakeholders report that oat is a really outstanding rotational crop. It provides a lot of benefits to the rotation, but production is really restricted by limited local markets and limited profitability. However, our research is showing that small grains, including oat, can improve the yield of other crops that are in the rotation that's shown for soybean in this graph. So the third component of our research program will be geared toward optimally placing oats within the rotation, both as a cash crop and as a cover crop. And we're particularly interested in finding the mechanisms by which oat contributes to the productivity and sustainability of the cropping system as a whole. And that's almost certainly influenced by soil biology. So that feeds right into our overall research program very well. And we think that by improving the dependability of oat production under varying field conditions and the quality of the grain that's produced, we can really increase the profitability of oat as a cash crop. And that's going to be key to increasing oat production in the US. We also think that by finding the best ways to capitalize on oat as a rotational crop, we can really increase the adoption of oat in the US and simultaneously provide some much needed diversity to cropping systems in the United States. And so we're really happy to be part of this research project. With that, I'm going to pass it over to Tanner Shen, who will address data management and integration.
Well, thank you. So today I will talk about the data management component of the Oat Grand Challenge. This part will be covered by Jean-Luc Jannink and me, Tanner Shen, for representing the grain genes and T3 databases. So next. So the important thing about data is that it is created, it is processed, and it's published. But then what happens to it? So when we are doing the research, we focus on the data life cycle and doing creating the data. But then we understand that the importance of data when we try to find a paper published 10 years ago. So we try to find the data points there, and we cannot find it. So the importance of then the storing the data and making it accessible and available to people are become obvious at that point. So we try to capture this data life cycle by providing an area to researchers. So we do this following the FAIR principles. So make it findable, accessible, interoperable, and reusable. Next. So Grain Genes is such a centralized place where we gather data. We have been around over a quarter of a century now, and we have been largely funded by USDA and also US Wheat and Barley Scheme Initiative. So our data is curated. So a curator, a PhD level curator usually takes the data and then integrates data with the rest of our systems. Next. At Grain Genes, we do have data for wheat, barley, rye, and oat. Today, I will just talk about the oat portion of things. So we do have tools, community resources, data, and tools. Next. So the oat newsletter that oat community is one of the central places is managed by the Grain Genes. Manage in the sense that we manage all of the infrastructure of the oat newsletter so that the community can access it easily. On the next, on the data side of things, we do have tons of data on genetic maps, QTL, and markers. And most recently, we are collaborating with AFC, especially with Charlene White, to curate this data into Grain Genes with the help of our curator, Victoria Blake. In December alone, 22 new oat genetic maps are curated into Grain Genes. And this is only a partial list. So if you go to our website and search for QTLs or maps, you will find a lot of QTL maps, QTLs and genetic maps. Next. This is one example of having several genetic maps in one place. You can go from one map, one QTL location to others easily through our tool called CMap. Next. And although we have lots of genetic data, we also have genomic data as well. For example, with the availability of the genome assemblies, right now we do have genome browsers. Most recently, this is the PepsiCo genome browser, where genomic features are expressed in one-dimensional lines. And this is actually a shout out to PepsiCo because PepsiCo not only sequenced and assembled, put resources to sequence and assemble the highest quality oat genome assembly, but it made sure to make it available publicly and freely. Next. So it is not just like a visualization, but what if you have a sequence and you want to know where it is located? So for this, you can use the BLAST tool. You can put your sequence in, and then you can actually look at your hits. And if one of these hits is on the genome browsers, one of the genome browsers that you will choose, then you can click on this link and go to the genome browser page. So this gives an integral view and more functional view at Grain Genes. Not only that, from the genome browser, you can go to different places. This includes Ensemble and as well as T3, which Jean-Luc Jannink will cover now. Next.
You're muted, Jean-Luc.
Yeah, sorry about that. I'm Jean-Luc Jannink. I run the Tradicity Toolbox, or my program does. Before I start, I want to say that I conduct my research on the traditional homelands of the Gayogohono people, the Cayuga Nation, and I acknowledge the painful history of Gayogohono dispossession, and I honor the ongoing connection of Gayogohono people, past and present, to the lands and waters around me. So it's the Tridicy toolbox, but we have an important oats component. We've been accumulating data for the past 10 years, but some of it is historical, so it goes further back. And all this to say that we have a lot of data that is relevant to oat production from breeder trials. So yield data, test weight, and so on, over a large geographic area that's relevant to us. So in effect, we become a sort of collaboration tool for the large unstructured group that are oat breeders in North America. And the data accumulation continues. Since the start of the Grand Challenge a year ago, we've added close to 400 oat trials. Next slide. So in addition to being a data warehouse that can combine data, we have functional tools that can help breeders to manage their programs, design trials, do inventory, capture phenotypes on mobile devices, evaluate and make decisions. So these are possibly tools that can help in breeding, in bringing better oat varieties to market. Next. So what's the point of accumulating all this data? I think Tanner mentioned it well in describing the life cycle of data. What we do by capturing it all together is we enable joint analyses of all of this data. And so what you see here is that we have a number of trials. I mean, you saw the numbers, sometimes several hundred. Many of those trials have accessions that were genotyped. And so we can run GWAS analysis. And that leads to what I'd like to think of as knowledge. So specific areas of the genome where we identify that there is a locus that affects a trait. And from there, we can go back to grain genes and look at this, the context of that GWAS hit that's provided by the rich JBrowse resource that is on grain genes. And so there are automated links that take you from T3 back to grain genes. Next slide. In addition, something that's important, I think, for oat producers and researchers is this idea of mega environments. So as I showed, we have trials that span large swaths of North America for oats, and you can analyze this data to identify where oat varieties respond sort of similarly and where they respond dissimilarly, and identify the so-called mega environments. So geographic, more or less contiguous geographic areas where you might expect similar rankings among a set of oat trials. And so this has been work in collaboration with Lucia Gutierrez at University of Wisconsin-Madison, who's taken a lot of this oat data from T3 and run sort of detailed genotype by environment interaction trials. So that's some of the uses of T3. And with that, I'll go to the next slide and pass it on to Jim Harnly, who's going to talk about oat nutrition data.
Good morning. I'm Jim Harnly, and I'm the research leader for the Methods Application Food Composition Lab. We're an analytical lab, and we also run the USDA nutrient database. So my role in this consortium here is to analyze the oat materials that my colleagues are developing. Next slide. The real key here, the analytical emphasis, is we want to look at the variability of the nutrient components as a function of the experimental factors. The variability my colleagues are introducing. Metadata, this is essential. We're going to look at the variability as function genetics, environment, soil management, and processing. What do we analyze? We're going to look at minimally thiamine, lipids, fiber, and minerals, but there are many other analytical aspects we're interested in. Standard is proximates, total protein, lipids, carbohydrates, ash, moisture, and you get energy from that. Then there's lots of vitamins, full minerals, sweet, soluble, insoluble fiber, amino acids, and then we do metabolomics and lipidomics. So you can get a lot of information. Some of this will be done on every sample. Some of it will be done on composites. Analytical methods we use, pretty standard, ultra high performance, liquid chromatography, high res accurate mass spec is really the tool of the trade for us these days. And the data processing. To get some of the correlations we want, we use classical statistics, chemometric methods. and artificial intelligence, neural nets, and so forth. Next slide. So this kind of sums up what I was referring to as the impact of the metadata. You can see at the bottom, genetics, location, soil management, these all influence the harvested plants. Processing then has another influence, and we get a final oat product. If we go to the right, you'll see the analyses. We either look for patterns, and Jay Baum described some of these already with near IR and mid IR. And we also look at targeted specific components. So with all these methods and all these analyses, we can get a pretty good feel for the characteristics of the plant materials. And then finally, you look to the left, we'd like to trace it down to soil management too. How are the changes that Sharon and her group are making on the growing of the materials affecting the final outcome? Again, another area for analysis with some of the chemometric methods. Next slide. This is kind of an example of some studies we did before, dry beans from Mike Grusak. This slide represents 300 genotypes at 7 locations and done with principal component analysis. The take-home message is location, location, and location. You can see that the 7 locations are pretty well clustered regardless of the genotype. But anyway, moving on, next slide. Our database, Food Data Central, consists of 5 databases, branded foods, 350,000 records. This is basically label data. Classic Standard Reference. This is our old database where we analyze composites. The Food Nutrition and Database for Dietary Studies. This data goes to NHANES at CDC for what we eat in America. The new ones we have are Foundation Foods, which are analyses of the single organism foods such as apples, potatoes, and so forth, and experimental foods. That's better defined there at the bottom. Cultivars not on the market, new analytical methods, new processing methods, relationships and composition not looked at before. And we hope, we will put in all the grand challenge data here. So we've got dairy data coming in. We're looking at cold environment agriculture, pulse crop health initiative. And eventually, in a year or two, we'll start putting in the grand challenge data for oats. So with that, thank you very much. Back to you, Jose.
Okay, thank you, Jim. Okay, so thank you, Jim. In conclusion, this Oat Grand Challenge project is a massive collaboration involving many USDA scientists who are collaborating to help achieve the goal of expanding oat cultivation through higher yield, enhanced disease resistance, higher quality. Also look at production practices, and as Sharon pointed out, profitability should be a key here, and enhanced nutritional quality. Also, I wanted to point out here the great support from our stakeholders who have been key in supporting this research, especially Oats in ARS, especially NAMA, the North American Millers Association, for their support that has resulted in almost $4 million added permanently to the USDA-ARS Oat research budget in the last 3 years. And these funds have been spread out into Aberdeen, Idaho, Fargo, North Dakota, St. Paul, Minnesota, Brookings, South Dakota. And the bottom line is that of all these new funds that will help with this research, also we will have, as I mentioned earlier, a new oat breeder in Fargo, North Dakota. Next slide. So we are not doing this alone, and I want to thank first all the members of the ARS team that are listed here. Of course, we have great support from universities, as were mentioned during the talk. I tried to list them all here, and I may have missed a couple. So we have great support from university scientists and professors, also from the industry. You heard about PepsiCo sharing data, and others, Corteva, and then also international collaborators have been great, and we're leaning on them also to help oats in the US. Next slide. Next slide. So I'll open up for questions here. Can you go back one? So here is a picture of the US uniform nurseries planted in Ottawa back in 2014. And here's Howard Rines, as you heard during the talk, that he was recognized for the genotype that we're now looking at transformation. Also Art McElroy, a Canadian breeder there. So I'll open up for questions now. Thank you.
Thank you, everybody. Thanks, Jose, and thanks so much to all the speakers. That was really great. Sorry, my fingers have slipped on a few of the slides, and I haven't caught up on the Q&A box. So let's start with some of the questions that were actually answered through text, but I'll read them again anyway. So Steve Harrison has asked Jason, how close are we to an effective genomic selection platform for oat disease traits, particularly crown rust horizontal resistance? And Jason replied, the Northern US Canadian breeders are all in various stages of developing and using GS in their programs. Crown rust resistance is one of the traits they select for. I think a meeting with all the breeders might be useful now, particularly as to develop new GS-focused genotyping platforms. And Jason, did you want to add anything else to that, or?
No, I guess not. But I think it would be a pertinent idea right now to try to get all of the breeders maybe together to kind of talk about what we want to do for genomic selection moving forward, how we want to do it, and if doing something centralized out of the genotyping lab makes sense.
Okay.
Can I jump in? Can I jump in, Charlene?
Sure, sorry, I'm just also
Yeah.
going to stop sharing my screen because this is difficult.
That's fine. I just want to mention Bruce Roskins. Recognize the contributions of Bruce over the years. He's been fundamental in getting support in ARS for oat research overall. So happy birthday, Bruce, and thank you.
Thanks indeed. And yeah, happy birthday, Bruce. Okay, so another question that was answered, but I'll repeat here anyway. Jennifer Mitchell-Fetch is asking, Shahryar, are the QTL you found adult plant resistance genes or genes with all stage resistance or both? And Shahryar replied, we know that individually these genes, that is single QTL, do not provide great seedling resistance to a specific isolate, but provide adult plant resistance in the buckthorn. When combined, it seems we get much better seedling resistance and additive effect on adult plant resistance. Not sure if this answers your questions, but I would say when combined, both. Shahryar, did you want to add anything to that?
No, I think that says it all. So we're good. Unless Jennifer has a question to follow up.
She'll have to type it if she does.
Okay.
Okay, so there are a number of open questions here. Bruce says, if there is agronomic response data available on advancing oat lines from the USDA uniform nurseries, could that data be included in those annual reports? That's the uniform early and mid-season nurseries. It could provide better guidance as to the best G/E deployment of those new lines as they move to registration. Who would like to answer that question?
Wait, Bruce, can you just give a little more detail on what you mean by agronomic response data? Obviously, the reports have yield data or whatever across all of the different locations. So what exactly are you looking for?
He can't answer live.
Oh,
right
he can't
now.
answer live. Oh, I'm
Unless,
sorry.
Grant, you could add Bruce Roskins to that.
I've just added Bruce as a panelist. So Bruce, if you're with us, you can unmute and speak.
I mean, I suppose we
Okay,
could try to identify. Yes, we can hear you.
Okay. What I'm referring to is I'm really enthused about the work that Sharon's doing on South Dakota, looking at some of the different agronomic responses to the oats. And since, you know, one of my issues over the years is we do not a very good job of introducing the new oat varieties that are coming through registered by the various state crop improvement associations, but in a lot of locations, they're just out there. We don't have any recipes, and I've overused that word. We don't have any good recipes for how to best incorporate those new lines into a farmer's production. And I think with the work that Sharon's doing out there, saying that this variety works better under droughty conditions or not, would give a better indication to predict, particularly a seed company if they're interested in that variety, but more particularly to the farmers, rather than just looking at, okay, it didn't do real well in Minot, but it did okay in Fargo. I just think the more agronomic information we can put into the release of some of these new lines, the better they would be utilized. That cleared up for anyone?
Yeah. So you mean actually like response to management, not just response to location by year environment, but possibly nitrogen rates or something like this. If I mean, I don't know if that data is available.
Well, as I understood it, some of the work that is going on at Brookings is looking at some of that fertility, by some of those new advancing lines. I'll throw one out, one of them that came out from South Dakota State more recently, works better under organic conditions than it did under conventional conditions and stuff. So that type of information would be, we delved into some of the background work that Melanie had done and said, hey, this thing may have a future there, but probably under high nitrogen, no, it wouldn't work as well. So the more information we've got on that at release time, I think would be very beneficial.
Right, and for us, this is a new component of our research program. We've got a lot of research on crop rotations and interactions with soil biology and how different components of the system work together. But when it comes to specifically addressing oats in a special way, this is new for our lab, and so we don't have a lot of data yet.
And Catherine Howarth has just said here too in the chat that to follow on from Bruce's point, dissecting the involvement of genotype by environment in trait expression is important and presumably is covered in this initiative with your work, Sharon. So
yeah, we've only got the one guy, and he's really good, but
right, that's great. Thank you. So next we have a message from Juan Arbelas, who is introducing himself. He has taken over the Oat program at University of Illinois, is happy to meet us, and it has thought this presentation is extremely useful. In fact, maybe we could add Juan as a panelist to say hi to everybody so we all know who he is. He does have a question though. Kathy, you mentioned there's only one elite background being used for QTL deployment. Are there any plans to include additional elite backgrounds, for example, based on elite lines that perform well in certain mega environments?
Yeah, the one elite background that was on my slide is for mapping purposes, but I have also been making crosses with other elite lines that I have as I go along, and so that I have a head start on deploying those QTL in other backgrounds. And I would be interested in receiving material that people are particularly interested in having adult plant resistance QTL put onto.
Great, thanks. And Catherine again here, this is a great initiative. Is there a website associated with it with details of all the partners with contact details?
I would say not yet, but it's in the works. Thank you.
That would be something very useful.
Yes.
And Juan has joined us too, Charlene.
Oh, good. Yeah, I didn't see him there. Sorry, Juan, where are you? Would you like to say hello?
Hi, everyone. Hello, I'm Juan Arbelas. I just recently joined the University of Illinois. I took over the oat breeding program, and I'm very excited to meet the oat community. This presentation has been amazing. So thank you so much for putting it together. I'm learning a lot. I have a lot of questions, so I will probably be posting them in the chat or email you personally. So thanks a lot.
If I can brag just a bit, Juan got his PhD at Cornell, and I can vouch for him. He's excellent.
That's wonderful. Welcome, Juan. Okay, so let's see. Tyre Wiesner-Hanks, prairie provinces have also seen oat acreage displaced by crops like corn and soy that can offer better returns. Yes, we certainly know that. What will ultimately make food-grade oats a more profitable choice for US growers? More competitive yields, better prices from the rising popularity of oat food products? Yeah, I got to knock corn and soy off there.
Well, I guess my presentation mentioned those things. I don't know that I'm really best qualified to answer them. Maybe we should put Bruce back in there or somebody. But what I hear from my stakeholders is that the dependability of quality grain production, they can usually get harvestable yield, but then they don't always know what the market value of that grain is going to be. So, a sliding scale of returns based on the grain quality, both in milling quality and in protein content and things like that. But just the low value of oat grain. It's a low input crop, which is good on the cost side, but on the return side, there's just a low grain price. And so increasing the grain quality would be helpful. And that's why what we're hearing from our people is that putting it into the cropping system and having it improve the profitability of their other crops from a number of standpoints, disruption of weed and pest cycles, improving soil quality, all of those things translate into improved productivity as a whole. And that's what's going to lift oat, I think.
Great, thank you, Sharon. I just realized too that Patrick Ewing is actually one of our panelists and he's new. Would you like to show yourself, Patrick, and just say hello? I'm surprised.
Sure, yeah. Sorry, I didn't realize that I was allowed to do this yet. Yeah, no, and I think that Sharon's point is exactly right, especially about quantifying and characterizing these broader cropping system benefits and looking at whole oats as part of this bigger system that could be more profitable with them than without them. That's I think the strategy that we're taking to complement your excellent efforts in developing more productive lines.
Thanks, and welcome to you too, to the oat community. All right, so different tack here now. Jaswinder Singh would like to know, what is the best way to estimate lipids in oat? Presumably aimed at Jay.
Yeah, I'm Jay at the wheat column at Fargo, and we also evaluate the total lipid content in oat, and we just determine the crude lipid content in oat. So I'm not sure whether our method is the best way to determine the oat content or not, but we use just hexane to extract lipid and then determine the weight of the extract oat after dry. So we currently are just determine the crude lipid contents, and also we tried to develop the NIR calibration models to determine the crude lipid content in oat, and usually the NIR model is working very well. So usually the calibration model shows over 0.9 R-square values. So I think it is good for screening the breeder samples. And we also used to have the use the GC to determine the fatty acid compositions, but currently we are not using it. But if you want to see more in detail, maybe you can use the GC methods.
This is Jim Harnly. Yeah, what Jay Baum has described is, in fact, the total lipids extraction. Hexane is a standard method. Fatty acid are analyzed usually with GC the fatty acids are extracted and modified, esterified, and then separated by GC. And then there's a whole brand new area these days called lipidomics, and you can literally pick out hundreds and hundreds of peaks, everything you wanted to know. And there's the polar lipids as well as the non-polar lipids. And how much information do you want? I think would be the ultimate question, or how much information can we afford to analyze? Anyway.
Thank you, Jim. Yes, I should have
mentioned. How much does a lipidomic assay cost? How much does a lipidomic assay cost?
It depends on what you do it commercially, or you can talk us into doing the analysis for you. And it also depends on how many samples too.
There you go. Yeah. So everybody be nice to Jim.
Yeah, it's difficult to put a number on it. When we get into the projects, I usually think in terms of what portion of a postdoc year would go into the project. So.
That makes sense. And the last question I have here right now is from Juju. Is there any specific gene or genes targeted in oat gene editing projects? So David.
Turn off my space heater in my freezing cold office. We don't really have a particular set of genes right now that we're targeting. My program is generally focused on trying to go after perturbation of disease susceptibility genes. And we're using brachypodium as a test bed for that. And that is a piece of cake to edit genes there. We're doing, I bet that's gone really smoothly until we have information on how well and what the effect is on knocking out different putative susceptibility genes. We won't know what we're going to try to export into oats. But you can imagine that for oat improvement, there's a whole spectrum of uses. Say from GWAS, you might be identifying candidate genes, and gene editing will allow you to validate those genes. There are traits in oats that you may want to modify. My background, back in the day was in metabolic pathway modification. So you can easily go in and try to, for instance, modify fatty acid composition by knocking out particular points in fatty acid biosynthesis, tocol biosynthesis and accumulation. That's all well established. We some years ago, my postdoc, Juan Gutierrez, identified all the genes involved in tocol biosynthesis. So there's, I think, a whole range of possibilities for gene editing. Again, the thing is that there are other traits that just are funky in oats. Naked oats is something really interesting, right? But it's associated with this sort of multiflorous trait, which is really not desirable at all, but it may be possible to go in and come up with another way to generate naked oats through information that we have from wheat and barley and the genes involved with that. So.
Great, thank you. See, Juan has indeed asked a number of other questions, some of which have been answered by text, but the one that's still open that a lot of people may very well be interested in. David, is the sequenced line OT3098 recalcitrant to tissue culture too?
We haven't tried it. And I think what we're really trying to do is just basically get all the parameters established with a genotype that does seem to regenerate very well. I don't think it'll be a problem to move into that genotype if, once we have our conditions worked out. Having said that, we're going to be having a flood of, I think, genome sequence information coming down. And so I don't really think it's important that that particular genotype be transformable and regenerable, right? We'll have enough information to be able to look at sort of particular genes that we're interested in and be confident. I mean, it's pretty simple to actually just re-amplify through your proposed target in the genotype you propose to use and see if the target and PAM site are conserved. So I think just having, some of that raw sequence information is enough to kind of allow us to launch off into gene editing in a manner that doesn't really depend upon genotype per se.
Thanks. And Nick Tinkers reminded me that actually our group at AAFC and McGill University are in fact sequencing Park. So that sequence will be available hopefully sometime soon. Also, this does kind of segue into our next Speaking of Oats seminar, and we are over time. So Jim, Bridine, what do you think we should do here? Should I go through Juan's other questions, or we can also post what's here in the Oat newsletter as well, the written answers?
Yeah, I would suggest that everyone has access to the answered questions. So if you go to the Q&A box under the answered tab, you'll see those texts. But if there's a way to summarize in the Oat newsletter, anything that is of broader general interest, I think that'd be great.
Yeah, I can do that.
And I want to just echo some of the comments we're seeing in the chat that this was really an excellent set of talks. Thank you all for your efforts in pulling all this together, but especially for the hard work you're doing in Oats. It's really uplifting to see this much energy and brain power working on this crop. So thank you. And Charlene, do you have a closing slide that announces the next Speaking of Oats?
Yes, I do. And I've just messed up my system. So I am trying to.
Well, I could share it's our next Speaking of Oats will be on April 15th. And that'll be at 2 p.m. Eastern time. So it's a little later. And we'll hear from Jorge Dubkowski, Alicia Del Blanco, both from UC Davis, and from Jaswinder Singh from McGill University. So it should be a really fantastic group. Grant Zadzik is working on the sort of the back end, all the technology, and we'll announce that very soon. So watch your email and the newsletter for registration information. And thank you all for being part of Speaking of Oats today.
Thank you.
Thank you, everyone.
Thank you.
Thank you.